This is lecture 5A, part 2. So the blood pressure is the key parameter that's regulated in the vascular system, and an important part of that also is blood flow. The pressure, of course, is the force that the blood is applying on the walls of the vessels, and it decreases so that when it by the time it gets to the capillaries, it's low enough that it doesn't rupture the capillaries, which are very thin-walled. So in an individual capillary, it is quite low, and that's mainly due to the fact that you have to consider that an arteriole comes in with a fairly high pressure blood and then divides into quite a wide network of capillaries, and you have to take into consideration the total cross-sectional area of all of those capillaries. That total cross-sectional area is much larger than the arteriole that fed blood into that, so the blood pressure drops. The volume, cross-sectional area is much bigger, volume is bigger that it's going into, the blood is entering into, so the pressure drops. The blood flow rate slows down, and that allows enough time for exchange to occur at that level, at the level of the capillaries. So if you look here at this diagram, which is showing in this area here, the largest arteries and smaller arteries, and then arterioles that feed into capillaries, and then those go to venules and veins. You see that the blood pressure drops off significantly at the level of the arterioles, and by the time it gets to the capillaries, it's really quite low. It's enough to provide enough hydrostatic pressure that adequate diffusion can occur. And then in the veins, in the venules and the veins, it's quite low. In fact, lower than in the capillaries. The total cross-sectional area, as you can see, rises quite a bit, and that accounts for the, in this area from here to here, and that accounts for the fact why the blood pressure is dropping so much over that span. The other thing, of course, is the velocity or flow rate slows down. So in terms of the arterial blood supply, the side, in terms of the arterial side of the blood supply, the arterioles are the smallest of the arteries. They have muscle fibers wrapped around them, which act as sphincters. In fact, they could be called sphincters, really. It's smooth muscle. It's not a continuous layer of smooth muscle at that level of the vasculature as you had at the artery level, but in different areas you have smooth muscle fibers wrapped around, and they allow the diameter of the vessels to be carefully regulated in response to signals from the autonomic nervous system, either parasympathetic or sympathetic division of the autonomic nervous system, and thus control blood flow into a particular capillary bed via relaxing, causing vasodilation, or contracting, causing vasoconstriction. And here you see a arterial blood vessel with smooth muscle bands of smooth muscle around it. And here you see a stylized, I mean, it doesn't really look like this. It's many, many more channels in a capillary. This region in here is supposed to be all capillary. So this is, you know, the artist drawing something to give you the idea, but it should be more channels. But you have sphincters around, quite a few of them, so you could actually, in fact, bypass a lot of the capillary bed if you wanted to, And that's what's shown towards the bottom. There are two examples here. They don't both happen at the same time, and what they're showing towards the lower end of this diagram is with those sphincters closed. So most of the capillary bed, in fact, is closed off, and you're not getting blood flowing through it because of these, I'll mark them, because of these sphincters here and here and here. And so that's with vasoconstriction, you get a diversion of the blood flow. And they're saying, okay, so when it's open, it's flowing through, and that's in the upper part of the diagram, in all of this area up here. Okay, but normally it would be both would be shut off, or both would be open, or both would be partly shut off or partly open. When they're shut off, it doesn't mean that the blood doesn't flow through over to the venous side, because you have, you should note, you have this thoroughfare channel, which is open and allows blood to flow over to the venous side. So in this way, because of this ability to vasoconstrict or vasodilate, blood flow can be directed in a very precise way. So this is a way of allowing more blood or less blood into a particular part of the body. Regulatory systems shunt blood to specific vessels in specific locations. For example, muscles receive more blood during exercise. Intestines receive more blood after a meal. And regions of the brain that are engaged in particular nerve activity will also receive more blood. Similarly, the opposite is also true. Muscles that are not exercising receive less, not zero, but less. Intestines receive less if they're empty, and regions of the brain will receive less when they're not as active. Well, a quite important aspect of the vasculature, something which people don't hear about too much. So you always hear about gas exchange or nutrient exchange, but probably not familiar so much with fluid exchange. There's actually quite a significant amount of fluid exchange between the blood and the tissues, and it's it's all determined by blood pressure levels and also the osmotic potential of the fluid in the capillaries, in other words, the blood plasma. The osmotic potential of the blood plasma is its ability to cause water to diffuse in towards the plasma. So, there is quite a bit going on, and there's quite a actual important balance, and it has to remain regulated carefully. Otherwise, you get a inappropriate imbalance of fluid distribution in the body. The fluid compartments, I would remind you, are intracellular fluid and extracellular fluid. And the extracellular fluid can be broken down into two parts. One is the interstitial fluid, which is between cells in the tissues, and the other is the intravascular fluid, which is inside the blood vessels. In other words, the fluid part, the water part of the blood plasma. The ability of any particular molecules to cross the wall of the capillary depends on these three factors: the capillary architecture at that point in the body, it can vary, as you know now, the type of molecule we're talking about, and the concentration of that molecule between its distribution on either side of the wall of the capillary, between the blood and the interstitium, or the tissue space. Well, here you see a diagram showing a capillary bed here, and the fact that it's surrounded, in this case now they've drawn in all the cells that the capillaries are spread out through. And what you see here in this diagram is that molecules are moving outwards here, and this side is the arteriolar side. So this part of this diagram represents this here, and on this side we have the venous side of, or venular side of the capillary bed. Now, in terms of movement of fluid towards the arteriolar side of the capillary, you have net movement outwards, as shown by these arrows, and moving as the blood progresses through the capillary and goes towards the venular end of the capillary, and then fluid is pulled back in. Okay, and this is shown in this diagram and is due to changes in pressure and osmotic potential. As a result of and you can see that osmotic potential is actually rising as you progress through the capillary. That's because water is moving out, and larger molecules that have osmotic potential and have the ability to pull water into the capillary are becoming more concentrated because of the water going out, and so the osmotic potential rises and water gets pulled back in. Essentially what this is pointing out to you is that there's a constant exchange of fluids between the capillaries and the tissues and back. And it's actually a little more complicated than that, and an important aspect is that not all of the fluid, water, that leaves from the capillary is pulled back into the capillary. Some of it stays, and we'll talk about that in a bit. Here you see the forces that are involved in movement of fluids from the capillary bed, from the capillary and into the tissue, and then back into the capillary. And depending upon what source you look at, you might see slightly different numbers, but in the end, at the arteriolar end of the capillary, all of this is capillary. It's all referring to capillaries, not to arterioles. The arteriolar end of the capillary, or the venular end of the capillary, at the arteriolar end, there's a network outward force pushing fluids out. And at the venular end, there's a net inward force pulling fluid in. This is the blood hydrostatic pressure pushing outwards. This is the blood hydrostatic pressure further down the capillary towards the venular end, and it's reduced because, you know, some of the volume has been pushed out into the tissues. Meanwhile, this is the colloid osmotic pressure, which is a force that's pulling water inwards due to the ability of large molecules, particularly a protein called albumin, which is far too large normally to leave the blood circulation, to exit. It has the ability, it has quite a strong colloidal ability. Some called colloidal osmotic pressure, sometimes is called oncotic pressure also. I'll write that for you. You might come across this word, oncotic pressure. So it just refers to the fact that there is a ability to pull fluids back into the capillary, and this causes osmosis, the diffusion of water inwards. So it's a ability particularly of molecules like albumin. Albumin is the most common protein in the blood plasma. It makes up about 50% of the blood plasma protein, and it's a fairly big molecule. I would point out that the colloid osmotic pressure here is the same in both on both sides in this diagram, but in the previous one they were saying that there was more pulling of colloid. In the graph on the previous slide, they showed that the colloid osmotic pressure actually rises, and so I would expect this actually to be higher than at the venular and then slightly higher. The end result is there's a hydrostatic pressure pushing fluid out, an osmotic pressure or oncotic potential pulling fluid in. Net outward force with these numbers that they're providing is 15 outward. Here the hydrostatic pressure of the blood is lower, 16, and osmotic pressure is 25 inwards, so it's minus, maybe even more, slightly more than 25. So there's a net inward force of -9. These two values don't balance, so some water is left in the some water that exits by osmosis and mostly due to the hydrostatic pressure remains in the outside of the capillaries. Now that water does not accumulate, otherwise you would get fluid accumulation and swelling, which is called edema. You'd get swelling of the tissues due to accumulation of fluids, and that's called edema. Instead, what it does is it drains into the lymphatic vascular system. You have a completely separate vascular system called the lymphatic vascular system, which can drain any fluid in the interstitial space. So interstitial fluid will move in. It only goes in one direction, back up towards the chest, and in the upper chest it enters back into the bloodstream. So it returns that fluid back to the bloodstream. Flow is unidirectional from fluid in the interstitial space into the lymphatics, and then flowing through a series of increasingly large vessels until it gets back into the blood in the chest, in the upper chest. Along the way, there are lymph nodes positioned all along the lymphatic vessels, and these are they're encapsulated, small encapsulated lymphoid organs that contain all of the immune cells needed for an appropriate immune response. In this way, you can you should realize that the tissues of the body are constantly being flushed, essentially. There's a flow through of fluid coming from the capillaries, going into the interstitial space, and then entering into the lymphatic vessels and traveling along through those lymphatic vessels, occasionally coming in, entering into lymph nodes where it can come into contact with a lot of immune cells. The thing about this flushing mechanism is that it provides a mechanism so that any foreign molecule, any foreign structure or particle that gets into the tissues will shed molecules from itself, and those will be flushed into the lymphatics and end up coming into contact with immune cells in the lymph nodes and triggering an immune response. So it's part of the protective system, this flushing mechanism of tissue fluid coming from the capillaries into the tissues as interstitial fluid, and then going into the lymphatic vessels, becoming lymph, and then flowing through. Here you see another diagram from another source showing the same idea, slightly different numbers. The numbers aren't, fixed in stone. There's a range of different numbers that you might see. Here, instead of 40, you see 32. Instead of 25, minus 25, you see minus 22. But you have a net outward force. And here, hydrostatic pressure is lower, 15. They've used the same osmotic potential, minus 22, and you have minus 7. The key thing is that 10 is greater than minus 7, so net overall there is more fluid going out than fluid coming back in at the vascular or venous end. And here it also shows that the fluid that doesn't return ends up going into the lymphatic vessels. Okay, let's talk a little bit about blood flow overall. What you have is a certain volume of blood flowing through the system over a given period of time, measured in milliliters per minute. In 1 minute, the amount of fluid going through is equal to what the heart puts out. It is a closed system, essentially. So the heart pumps out a certain volume of blood, and it's determined or calculated per minute, and it's called cardiac output, or CO. Cardiac output is the volume of blood that the heart puts out in a minute, calculated by multiplying heart rate per minute times the stroke volume. At rest, it's relatively constant. It could vary between different organs. It could change. If you've just eaten a meal, more blood might flow into the intestines than elsewhere. But so it is redirected at times, but in terms of overall at rest, cardiac output is going to stay the same. It's pretty constant within a range. But as soon as a person becomes active, then things change. The blood pressure, in fact, is the force per unit area on the wall of the blood vessels. It's expressed in millimeters of mercury. And like all fluids and gases, the blood will move or flow from according to the pressure gradient. It will always flow from high pressure to low pressure areas. So the differences in blood pressure within the vascular system provide a driving force that keeps the blood moving constantly from the higher pressure area to the lower pressure areas. And you saw in that graph of looking at the distance away from the heart, first at the on the arterial side, and then in the capillaries, and then on the venous side in terms of distance from the heart, and pressure. You saw that the pressure, once you start getting into arterioles, starts to drop and is quite low in the capillaries, and then very, very low on the venous side. So there is a driving force, there is a pressure gradient, and the blood keeps moving back towards the heart, aided on the venous side, of course, by the factors I talked to you about, mostly respiratory activity, respiratory muscle activity, and skeletal muscle activity. Now, in terms of flow, one of the things that will resist or oppose flow is resistance. And it really reflects the friction that the blood encounters as it goes through the vessels. And this is mostly, we're looking here at the systemic circulation, not the pulmonary. And this is referred to as the peripheral resistance. So we're talking about peripheral resistance, or PR. We're talking about the overall resistance on the systemic side of the circulation. There are three key sources of resistance that overall make up peripheral resistance. One is blood viscosity, the thickness of the blood. The thicker it is, the more resistant it is to flowing. The other is the total blood vessel length, and the third one is blood vessel diameter. It shouldn't take you too much to realize that viscosity does not radically change hardly at all with time, nor do you keep growing to any significant level lots of new blood vessels, so the total blood vessel length doesn't change. The thing that can change, and therefore can alter resistance, is blood vessel diameter. All right, vasoconstriction, vasodilation. You can get more resistance to flow in a particular area of the body, Or you can get overall more resistance to flow if you have generalized systemic vasoconstriction causing, the spaces to narrow. So these things are relatively constant, the thickness and the length, the viscosity, in other words, and the blood vessel length. But vessel diameter, the degree of vasoconstriction or vasodilation, does change. And it changes in response to the requirements of the body and signals coming from the brain to the smooth muscle that surrounds the blood vessels, particularly the arterioles. Now, resistance in this case there's a huge effect of vessel radius on the degree of the amount of resistance to flow. The resistance varies inversely with the fourth power, the radius to the fourth power. So any small change in radius, any reduction in radius causes resistance to rise dramatically. Similarly, any increase in resistance in radius causes resistance to drop radically. They are inversely proportional to the fourth power of the radius. As an example, if the radius is doubled, resistance is 1/16 of what it was before. So with vasodilation and doubling of the radius, you get 1/16 less resistance to flow. In other words, flow will increase into that particular area. Major determinants, as I've said before, are the arterioles, the smaller diameter arterioles, the major areas where you get peripheral resistance in the systemic circulation. A problem a pathological problem that can occur that can reduce the diameter of a blood vessel are fatty plaque deposits due to a disease process called atherosclerosis, which occurs with age. Pretty much every single person, even young people, will have some changes that can be observed in their aorta with in a teen even, you could see there are subtle changes. And as people get older, they get more and more changes in the wall of blood vessels on the arterial side particularly, and you get more stiffening and sometimes buildup of fatty plaque, which is a problem because that causes turbulent blood flow. Most of the blood flow, or when things are working properly in a blood vessel, blood flow is laminar. You have very straight laminar flow. That means there are all these layers of blood following going from the wall and inwards, all flowing smoothly, not turbulently, through the blood vessel. But with some sort of accumulation of obstruction on the wall of the blood vessel, like a fatty plaque, you'll get a disruption of flow and it starts to change, just like a rock in a river. If you have paid attention to what happens with a rock in the river, and you'll get turbulent flow, and just on this side you'll get eddies, and the blood slows down and has a chance to interact with the wall, and slower flow and more chance, in fact, of thrombus formation. So with that deposit of fatty plaque with atherosclerosis, you can get resistance increasing due to the turbulence and the narrowing. Blood flow and blood pressure are directly proportional to each other, and the key driving factor in flow between 2 points is always the pressure gradient, or delta P, between the two areas. So what we have here is if you look at pressure in one area and pressure in another area, if the second pressure is lower, then flow will be in that direction. So you subtract and you can determine the difference, rather, by subtracting one from the other, you get the gradient, delta P, and flow will vary directly with that. The bigger the delta P, the more the flow, less the delta P, less flow. And in terms of resistance, blood flow is inversely proportional to resistance. So if resistance increases, blood flow decreases, and vice versa. In the end, flow, F, is equal to delta P over R. So flow, or F, flow is equal to the pressure gradient divided by the resistance. Okay. If we consider the overall systemic blood pressure, okay, that's in all the vessels. The heart generates that blood flow into those on the arterial side. It's ejecting blood, pumping it into the arterial side, and the blood will move along the pressure gradient, going from higher pressure to lower pressure areas, so it moves forward. And if there is resistance, then pressure will increase. If there's an increase in resistance, pressure goes even higher. Overall, the highest pressure is in the aorta, where the blood is first pumped out of the heart. That's where you get the highest overall pressure in the blood, and then it declines the further you are away from the pump or the heart, mainly because it has to travel through a longer pathway, and so there's more resistance, and with increasing resistance there's less flow and therefore less pressure. In the end, when you get all the way through the arterial side, through the capillaries, and through the venular side of the circulation, and you get all the way back to the heart, to the right atrium, the pressure in the right atrium during diastole is 0. Okay. The steepest change, or the steepest delta P pressure gradient, is occurring in the arterial. And here, and we saw that on the graph earlier, and here you see the pressure as we move along through the circulation from the aorta, just outside the heart, through the whole arterial system, through capillaries, and then through the venous system, all the way back to the heart at that point. On the arterial side, you get oscillation in pressures between systolic and diastolic. The pressure wave created by the contraction of the heart during systole, so that upward movement or upward increase in the value in the pressure level because of the contraction of the heart ends up meaning, and then the drop with the relaxation of the heart means that you have this you have this range and pulsing pressure between systole and diastole. And in the larger blood vessels that are not too far away from the heart, like in the brachial artery in the upper arm where blood pressure is normally taken, you have a range, say, between 80 and 120 somewhere. This is actually like 85, 87. So here, maybe here would be the kind of area where you would start seeing the kind of pressure that you have in the brachial artery. And that's where blood pressure is taken. And then it falls off, and eventually in the smallest arterioles, it's no longer pulsatile. It doesn't pulse or alternate between higher and lower with systolic, diastolic. It becomes a constant flow and goes through the capillaries and then back to the heart. Arterial blood pressure reflects two key factors in the arteries close to the heart. Their elasticity, which is mostly due to the internal and external elastic lamina, but also partly due to the smooth muscle that surrounds it. Now, when we're talking about elastic tissues and tissues that need to expand, then in physiology, typically we don't talk about elasticity and then stretching of that and then recoil. We talk about typically compliance. Okay, You talk to physiologists, you'll hear them talk about compliance. They love to talk about compliance when they're talking about respiratory physiology, but compliance also applies to cardiovascular physiology. The compliance is, in fact, the ease with which the tissue is stretched out or expanded. So it's the distensibility. Nobody uses that term. They use compliance. Compliance is the ease of expanding or stretching out the tissue. The other thing besides compliance in the arteries close to the heart is the amount of blood that gets forced into them at any given time. That is the second factor that is key to arterial blood pressure. So how elastic the vessels are, how much compliance they have. If they have a high compliance, a high ease of expansion, then the blood pressure won't be as high as if they're stiff, more stiff, and aren't as easily stretched out. Okay, so if they're more stiff and are not as compliant, then blood pressure is higher. And if you're putting more blood into them, then blood pressure is higher. Okay, these two factors. And again, of course, it's pulsatile anywhere near the heart. When we look at pressure, we talk about systolic pressure, the pressure generated during contraction of the heart, systole. That's the pressure exerted on the arterial walls. It can be measured by checking the blood pressure in the upper arm using the brachial artery. Diastolic is the pressure during the relaxation of the cardiac cycle. So when the heart is filling, it's relaxed, and that's the lowest. Pulse pressure is the difference between systolic and diastolic. But it's not so much actually the difference between systolic and diastolic that's key to getting a good idea of how well a tissue is being perfused with blood. The better measurement is a value determined or called the mean arterial pressure, or MAP. The mean arterial pressure is the best pressure determination that can give you an idea of how well blood is perfusing through a tissue. Mean arterial pressure is calculated using this equation. The reason for that is that the heart spends more time in diastole than it does in systole. And so the mean arterial pressure is the diastolic pressure plus the systolic minus diastolic divided by 3. In other words, the pulse pressure divided by 3. This is the best value to reflect how well tissue perfusion is and how appropriate blood pressure is. Capillary blood pressure is low, not as low as on the venous side, but low, 20 to 40 millimeters. A low capillary blood pressure is desirable. If you have any higher than 40, you could rupture the capillaries and get little bleeds in the tissue because they're very thin, very fragile. The low blood pressure that you do have in a capillary, that low hydrostatic pressure, and remember when we were talking about hydrostatic pressure in the diagram with the calculations, we were talking about, they were talking about 25 millimeters or 22 millimeters. Here they're talking anywhere from 20 to 40 millimeters. So it varies, depending on the source and actually the site in the body. But this relatively low blood pressure is sufficient to force a filtrate into the interstitial space. The fluid that moves out, after all, is a filtrate because it's a filtrate refers to any fluid that's been filtered. That fluid that moves out into the tissue space, the interstitial fluid, is essentially the same as blood plasma minus the proteins. It doesn't have the proteins in it that are in the plasma. They stay inside the vascular space. So it is a filtrate because the water moves out but leaves the proteins behind. So the proteins are being filtered and kept in. And it contains electrolytes, ions, etc. But it doesn't have the proteins. So it is a filtrate and gets forced in the interstitial space. And it is enough blood pressure, this level of blood pressure, to cause diffusion of nutrients and gases and hormones into the tissues. Venous blood pressure is even lower than capillary blood pressure. The maximum, it would be about 20 millimeters of mercury. That's why a cut vein will flow. Blood will flow out of a cut vein, but if you ever see a cut artery, it's going to spurt, mainly because of that pulsation and oscillation between diastolic and systolic pressures. Overall maintenance of the blood pressure, the main factors are the cardiac output, the peripheral resistance, and overall the blood volume, the overall volume of fluid in the system itself. All right, now we already know, or you should remember, that cardiac output, we talked about this before, cardiac output is equal to heart rate times stroke volume. The number of times the heart contracts or goes into systole per minute times the average volume of each stroke, of each ejected volume of blood with each contraction. That's the cardiac output in a minute. And cardiac output is related directly to blood pressure. The higher the cardiac output, the more blood pressure. But also the greater the peripheral resistance, the higher the blood pressure. And these two are key factors. Blood pressure equals cardiac output times peripheral resistance. This is a separate thing that needs to be considered, blood volume. If the volume of fluid in your blood vasculature drops because of a bleed, if it drops enough, then the pressure will be inadequate to perfuse tissues, and if you have too big a blood loss, you'll start not getting enough blood into different areas of your body. The perfusion will be insufficient. The mean arterial pressure will be too low to adequately provide enough pressure for flow into those areas, and you start going into shock and you start getting organ failure. It's a serious business. But here, these equations show you the interconnectivity between blood pressure and cardiac output. So again, cardiac output, really, you need a venous return. You have to have blood coming back because, you need a stroke volume. A stroke volume, I would remind you, is equal to end diastolic volume, the volume in the heart, in the ventricle at the end, in the left ventricle at the end of diastole, the end of filling, minus what's left in it at the end of systole, the end of contraction. That's equal to stroke volume. And stroke volume, and end diastolic volume really depends on venous return. The heart fills due to the blood that comes back to it, the venous return. There are also neural controls that can affect cardiac output, and also hormonal controls. In the end, cardiac output equals heart rate times stroke volume. A resting heart rate is controlled by the cardiac center in the brainstem, mostly the cardioinhibitory center. It is receiving inhibitory signals coming from the vagus nerve. These are parasympathetic signals that slow down the heart. It's also controlled, an important feature is stroke volume, of course. Now we're talking about cardiac output. So resting heart rate is controlled at rest by the vagus output from the brainstem. So parasympathetic output slows down the heart. Stroke volume is determined by how much blood gets back to the heart, and determining therefore end diastolic volume. And then under stress, if things change and you get stressful stimulation or sensory input for whatever reason, and stress can be, the sudden need to run or sudden, need to move or act, then the sympathetic output of the cardioacceleratory center of the brainstem will increase its output, causing an increase in heart rate and an increase in stroke volume, and therefore an increase in cardiac output. If you have sympathetic stimulation, I guess it's easy for people to imagine that their heart rate goes up, but why the stroke volume goes up, they're not always clear. Well, the reason is that the end systolic volume decreases with sympathetic stimulation in the heart. The heart contracts harder, and there's less left in the heart at the end of each contraction. So end systolic volume decreases. If end systolic volume decreases, then stroke volume increases, according to this equation. And in the end, mean arterial pressure rises. And here we see a diagram. This is the last slide of Lecture 5A, Part 2. And what you see here is a flow chart showing the changes that occur when there is a drop in blood pressure that's sensed by baroreceptors. Okay, and they send information back into the brainstem. So the drop in blood pressure is information is sent into the medulla in the brainstem, and that will reduce parasympathetic activity. Okay, and it will increase sympathetic activity. So parasympathetic signals coming from the brain are reduced and sympathetic signals are increased. With an increase in sympathetic signals, you get directly increase in the heart rate, and also with a decrease in parasympathetic signals, you get an increase in heart rate because you have less inhibition. So you'll get an increase in heart rate. The other thing that increased sympathetic output or activity triggers is increased contractility of the heart muscle, and therefore end systolic volume is reduced, thus increasing stroke volume. So on this side we have two things that will increase heart rate and one thing that will decrease, and another thing that will increase stroke volume. And because cardiac output equals stroke volume times heart rate, if these two increase, then cardiac output increases. The other thing that I haven't mentioned is the sympathetic activity is not just directly to the heart, but we also talked about the fact that you have a innervation to the adrenal glands, triggering epinephrine release from the adrenal medulla into the blood, and that goes around through the blood and gets picked up by receptors in the heart to trigger an increase in heart rate. The other thing is that with activity, I'm not going to get into this too much, but no, I'm not going to discuss that part. Okay, so that's it for the cardiovascular part, and in this last slide, to sort of bring it all together again and remind you about the cardiac part. But the vascular part of cardiovascular is very important, and it's important to understand it. And that's the end of Lecture 5A, Part 2.